Welcome to the second part of our presentations or our unit on protein synthesis, translation of the genetic code, taking the messenger RNA, which was created in the last unit in transcription, and now using it to produce the exact perfect protein is translation. Here's an example of a messenger RNA that we made in transcription from the DNA molecule in the nucleus. So your codon, and think of each three of these little letters as almost like a parking place. The whole thing is the M or messenger RNA molecule. But each three, starting from one end and going down, is a codon. Because the transfer RNAs, as we will see in a moment, only have three nucleotides long, so they need to plug in exactly the right order. For example, the transfer RNA that would plug into the codon here under the word codon would have to be UAC. So that's the only thing that can park there is UAC. And the transfer RNA that drags in the amino acid that is UAC carries one and only one amino acid. So basically, the DNA code creates the messenger RNA code. The messenger RNA code creates a parking spot for a transfer RNA to come in and park, dragging in its load, which is that special amino acid. Ribosome on the rough endoplasmic reticulum is where the messenger RNA attaches. Now, ribosome is the protein factory of the cell. And so the ribosome comes along and that starts the reading process. It stimulates the transfer RNAs to be brought in. And again, they only can be brought in the exact right order to park in the exact right parking stalls. The transfer RNA molecule arrives. It brings amino acids to the first three bases, the codon on the messenger RNA. The first three unpaired bases, the anticodon in the transfer RNA, link up with the codon. So here we go. The transfer RNA comes in, parks in the only place it could. No other transfer RNA could park in this place. And then the amino acid, in this case symbolized by the greenish square, is the only amino acid that transfer RNA hauls or carries or brings in. And the anticodon UAC parks, matches up with AUG. Once that's there, another transfer RNA brings into the second codon, and that anticodon, that transfer RNA will park in there. And if you look carefully, after the first three letters, it's the messenger RNA is GGC, so the next transfer RNA coming in, the anticodon, has got to be CCG. And in comes a CCG carrying a different amino acid because every transfer RNA carries a different amino acid. And that is, so basically the DNA code has said, hey, we need the green box first, and then we need the pink box. And by using these letters, it ensures the perfect order of this upcoming protein. And in place, and once they're in place, then something called a peptide bond forms between the amino acids. It's the bond that bonds the amino acids together. So once that peptide bond is formed, then the first transfer RNA becomes free to leave and moves off looking for another green square to pick up for a possible another parking spot farther down the road. Off it goes. And it's been released. Chemically, the peptide bond releases the first one and moves off to find another amino acid. And now we look UUA, the third one that would be called for would be AAU. So another transfer, the ribosome kind of slides down the thing like an old-fashioned typewriter, and another amino acid comes in, AAU, dropping off its green or yellow square amino acid. Peptide bond forms. The amino acid transfer RNA on the left is going to leave, and it continues on, and it keeps adding and adding in just the exact perfect order. Another one coming in here shows you GUC hooking up with CAU until the end codon, the stop codon, is reached, GUU, saying stop bringing in. There is no transfer RNA that'll park there. And another one comes. And here we have the last step. At the very end, the transfer RNAs leave, messenger RNA goes back into the nucleus to break up to be used again, and all that's left is the perfect protein made from many amino acids bonded together, peptide bonds, in the perfect order and number to make it this special protein, whatever protein it's supposed to be. If it were off by one amino acid, change an amino acid, even out of order, the protein would not be proper, and basically it would be garbage.